Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 164th seminar, and I call it a National Science Foundation Day. I have been traveling since last Friday, and I am a small town in Dolomite, northern Italy. Often I feel nature is greater than human, and to understand ourselves better, we may study the greater and mysterious nature, which forms many fields of study, including physics, chemistry, biology, etc. We have been trying to conquer or control nature by engineering sometimes. Nature shows something engineering cannot create. Still, engineering has achieved tremendously, making human life better, healthier, and safer. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Erik Pierstorff. He is a program director of SBIR STTR at National Science Foundation, covering biological technologies and pharmaceutical technologies portfolios. By providing visions and support for biotechnology applications and translation, he has helped advance the research field and biotechnology industry. I met him at an engineering biology research consortium meeting, and he kindly accepted my seminar invitation. Eric, thanks so much for your contribution to the research community and biotechnology industry, and the virtual podium is all yours now, and thank you again.
Thank you for that introduction, and thank you for inviting me here today. Hope it's okay. I'm going to share some slides.
Sure.
So hopefully you can see this okay. Let me know if you cannot.
Yes,
All
perfect.
right, terrific. So I'm going to be giving a little bit different presentation than you probably see here. I'm going to be talking specifically about the SBIR program, but I'm going to try to, at NSF, and I'm going to try to gear it to really focus on what the NSF program likes to fund, the stage of company, the stage of technology, and some of the do's and don'ts and best practices that you can do when you're applying to the program. So I'll skip a few of my slides in the presentation to try to focus on that. This is a nice opportunity for people who are looking to build off of an academic career. You may be a postdoc, a grad student, a faculty member that has a technology that may be available for translation. And that may be an opportunity to start a company and get it out the door and get some benefit to the society from whatever that technology is. So traditionally that we have about $230 million and we fund about 400 startups a year. And this can be up to about $2 million non-dilutive, which means you don't give up any equity in your company for funding of that technology. So some of the key tenets of what SBIR is at NSF is we focus on very early stage, high risk innovation in differentiating technology. We focus on very early startups and being very early catalytic capital. So if you have a lot of resources, you've been around the block, or you're having an incremental advancement of a technology you've already developed, not a great fit for NSF. We fund all areas of science and technology and engineering with a few small exceptions, and we want commercially focused companies. So the team that we have in the program here has all either been involved in startups themselves, investment, or other advising of startups. So NSF focuses earlier and at higher risk projects than most other federal agencies. So there are 11 agencies that have an SBIR program. Um, NSF focuses on addressing market risk, and also NSF is never a customer. So a lot of programs are mission driven, where they want to have a technology that's developed so that that agency can then buy it. NSF does not do that. So you must have a commercial opportunity that will support your company moving forward. and you get entrepreneurial support from the program as well as from the program directors that you work with. So just some outcomes that we've seen in the past, 10 to 11 years have been about 4,000 awards to startups. And there's been about 32 billion on follow-on private investment, and about 420 company exits. So there's been a lot of strong uh, success, from these companies that have been funded by NSF going forward. And these are just a few examples that you can see, both on the impact of the technologies they've had in different areas, as well as the financial successes they've had from raising money moving forward. And again, just to support, what I've been saying, um, the 96% of the companies that apply that have been awarded have fewer than 10 employees. And to give just kind of a counterpart to that, to be eligible for the SBIR program, you need to have fewer than 500. So that just shows the breadth of, range of different programs there are in the government and where NSF really fits. The very early stage, very small companies. Most of them have been created in the last 5 years, and a lot of them are never applied to NSF previously. So it's a stepwise process. The first step is a project pitch. If you are invited to submit your project, you then can submit a full proposal, and then you have a decision made on that proposal, usually within 6 months. So the project pitch is a really brief web form. You can submit it at any time. You can only have one under review at a time, and you don't need to have a company formed, to have a pitch submitted. But what we will look for is the technological innovation, the market opportunity, the main technical objectives and challenges you have in the company and the team. So this is a really good opportunity for you to talk about what your technology is, why it's a good fit for the program, because that's really the focus of the project pitch. You're not pitching to an investor here. You're saying, am I a good fit for the program? Because we don't want you to spend a lot of time writing a proposal if you may not be a good fit. So if it is a good fit, we'll invite you to submit a full proposal. It's not, we'll give you some explanation as to why, or we may ask for a little bit of extra information. And then you can submit another pitch, the next quarter, basically, but you can't keep coming in right after another. You want to give you some time, think about, why you might be a good fit. So what does NSF not fund? So it does not fund incremental or evolutionary development, assessment of an already developed technology, straightforward engineering efforts with little technical risk, basic science research, business development, market research, and sales and marketing. There are a few minor exceptions within that, where you can have small subsets of funds for doing some business development things, but that's basically what it is. So this shows you want translational research. You're building a product, so it's not basic research, but we also want it to be that high technical risk and leverage NSF funding to really move the needle on that, get you out the door to the next stage. NSF as a whole does not fund clinical trials. We don't fund anything related to Schedule I controlled substances or any specific therapeutic candidates. Drug discovery is still there, but if you have a candidate in hand and you're doing your assessment of that, there are other agencies that are better fit than NSF. Um, so, yep, I will skip over this one for now. It's a phased approach. So phase one is currently capped at 305K. Phase two, 1.25 million, and then there are some supplemental opportunities after that. There is no direct to phase two, so it's a little bit different from other agencies. But there is a new fast track opportunity with some strict eligibility requirements. So if you have any questions on that, feel free to reach out and ask me. So again, what do we look for? The impact, the technical innovation, the market pull, and the potential for scale. There are some eligibility requirements that you can find online, so I won't go over that. But this is a nice project slide that I like to talk about. So why may you be a good fit? You still have a lot of significant R&D to do to overcome a significant technical challenge. Someone who's skilled in the art may not be successful in coming with that. So this is the risk that we want to have there, because there could be a strong impact and a strong commercial opportunity going forward. So think about this as, NSF is going to help you, de-risk and demonstrate that proof of concept in phase one, and then build upon that to go forward. You should have a unique and defensible technical innovation that gives you a durable competitive advantage. And you want a company structured for aggressive commercialization of the new product or service. We're not interested in funding mills who keep coming back to the well or have a lot of access to resources. We want the NSF money, especially at phase one, to be extremely impactful and leverage that technology to be pushed out the door. And you want to have a good understanding of market and customer, indicating potential to hit an unmet customer need, customer discovery, market reports, anything that you can demonstrate that what you're doing is going to be wanted on the commercial aspect. And this is, again, since NSF program is not mission driven, funds all areas of science and engineering and technology development, you want to make sure there's a commercial opportunity. And that's going to be assessed even at phase one to understand it's like, why would we build if no one wants to buy it? So you want to really consider that as well. Um, so there's some additional information, the proposal review and decision generally happens within 6 months. Um, and if you want to engage, sign up for the mailing list. Go to the website seedfund.nsf.gov. There's a lot of great resources there about the process, the different registrations you'll need at different stages. And the first step of point of communication is always a project pitch. If you have your idea, submit your project pitch. That's the first step of communicating with the program. You can also reach out to me if you've seen this video. Feel free to reach out to me. I can point you in the direction of the project pitch or maybe answer some general questions. And I went quickly because I know that we want to do that and hand it off to Claire. But happy to answer any questions that may come up as well.
Fantastic, thank you. So one thing I want to add is many people, especially some, basic science department never thought about, commercialization, but I wanted to say real breakthrough technology coming from real fundamental basic science that is kind of change entire human, society, tremendous way. So bioeconomy is emphasized for many, many years and then gonna be emphasized for next, you know, 10 or 20 years or more. I believe, anyone, doing even basic science also potentially considering, this program because of money is a lot actually compared to other NSF program in general. So thank you so much. I mean, this is so exciting. Also, this is very important for me to remember NSF emphasize more innovation than other agencies. That's so fantastic. That is kind of more for the professors, I guess. Thank you.
Of course. Thank you so much.
All right. Now, the main speaker of today with a longer introduction. Dr. Claire McWhite is an assistant professor at the University of Arizona in Tucson, where her group studies how functional information is captured by protein language models. You may say, I mean, she is now in very hot Arizona, but I found out, today she is also in Italy, northern Italy, Milano. It's only 4 hours from me, and I would say what a coincidence or interesting kind of, the fate. She received her PhD in molecular and cell biology at the University of Texas at Austin, supervised by Dr. Edward Marquardt, where she worked on high throughput discovery of conserved protein interactions. She was previously a Louis Singler Scholar at Princeton University, where she was advised by Doctors Joshua Akey, Mona Sin, and Martin Jonikas. Claire, thank you so much for your time today, especially, you probably want to go outside to enjoy the Italy, but doing this one for us. And please take it away, all yours now, and thank you again.
Thank you for the introduction and the biography. Let me just share my screen. Okay, so the slides are visible. Okay, so for today, I'm going to talk about the language of proteins. So I broadly work on the sequence to function question, which is given a protein sequence, what can we predict that it does? Let's see. Oh, there's been a lot of progress recently in the past few years on the sequence to structure question, which is, given a sequence, what's a possible 3D fold that that protein can have. However, what's been much more difficult has been the sequence to function question. If we know just the sequence, what can we say about that protein? So in terms of function, I'm thinking about things like protein interactions and how is this interaction specificity controlled through the sequence? We have variant effect. How do the mutations to that protein affect the protein function? And also looking at functional divergence, say when our two proteins have a similar function and when do they have a diverged function? And what are the mutations that account for that divergence? So we can think of protein sequences like a language. If a language is a series of tokens where token meaning depends on the context of each word. So in language, we have I threw a coin in the wishing well, or I did well on the exam, and the meaning changes based on the surrounding words. And it's the same thing for arginines in a protein sequence in this case. Each of these different arginines is going to have a different role within the fold based on where they are in the 3D structure. Now, language models are capturing the syntax or the sentence structure of human language as well as protein language. By syntax, I mean specific relationships between tokens. In normal language, we have relationships like subject-verb relationships or adjective modifier relationships. But in protein language, the main syntax feature are these motifs. So for example, the zinc finger motif composed of these two cysteines and two histidines. So I want to give a bit of intuition for how language models are made. One method is to predict hidden amino acids. And so what's happening is this model is learning to use amino acid context to predict the identities of hidden amino acids. So inaccuracies in this prediction are how the modeled weights are learned over looking at thousands and millions of sequences. And so what we're doing is in our input sequence, for example, hiding this arginine. And then the task of the model is to predict what that should be. And errors in this prediction drive the model parameter tuning. As a kind of conceptual example, we can think of an input sequence being the cat meows. Maybe the model then sees the cat hidden word or the hat. And then the first guesses are going to be random, like the cat philosophizes, the cat lamp, the cat Milan. And then parameters are updated until we start to get correct answers, like the cat meows, purrs, sleep. And this is how it also goes with proteins. Models capture what is permitted according to the structure of the language that it's learned. And protein language models are quite complementary with traditional phylogenetic techniques. So if we think of a protein sequence going through a protein language, the protein language model is recognizing co-occurrence patterns, co-evolution, contextual motifs, which in the end is giving us these contextual numeric embeddings representing each amino acid, which we then summarize into a single representation for the whole protein. And we can use those numeric embeddings to, for example, calculate similarities between the function of different proteins. And this is in contrast to sequence analysis, where we're looking at sequence analyses, determining the distance between the sequences, and then assembling a phylogenetic tree. So critically, embedding similarity captures shared patterns within sequences and only indirectly correlates with the actual evolutionary relationship between proteins. On the other hand, sequence similarity from phylogenetics informs on evolutionary relatedness and sequence history and only indirectly reflects functional similarity. Because a single mutation in two closely related sequences can cause a large functional shift. And there can be two proteins that have relatively diverged evolutionary histories that have convergently evolved a pretty similar function. We can also think about mutations using the language framework. So a neutral mutation, equivalent sentence meaning, like I drank a cold coffee, I drank an iced coffee. It's a mutation and it's a change, but it hasn't actually changed anything about the meaning. I think of a deleterious mutation as a nonsense sentence, such as I drank an iced coffee becomes I drank an iced computer. It doesn't make sense. It's not expected according to the rules of the language. And then finally, the most interesting is a function changing mutation, which is a change sentence meaning. I drank an iced coffee or I drank an iced tea. This is a mutation that is still valid. It still forms a valid sentence, but it does change the meaning of the sentence. Corresponds to novelty in protein evolution. So when a language model encodes a protein sequence, we can measure which amino acids are predictive of each other. And so this is just to introduce the concept of kind of attention. So in the zinc finger motif, when you hide one of these C's or one of these H's, the surrounding C's and H's of the motif are useful at predicting that missing identity. And so you can see networks that reflect that these C's and H's are attentive to each other. And so what we're doing is going from this kind of one or two-dimensional view of sequence information that we can get from doing multiple sequence alignments to a new way of representing sequence, which is hundreds of attention networks of relationships between amino acids overlaid onto protein structure. So before I get into the data, just to kind of summarize this intuition section, protein language models are extraordinarily powerful tools for biology. For me, I use protein language models as tools to investigate specific biological questions based on the underlying idea that the protein properties that are controlled by sequence are captured in the complex and cryptic data structures of protein language models. And seeing what useful information can we extract from these models, and how can we use language models to approach problems of experimental and computational biology in new ways. So first, can we learn to see like a language model does? So this is a protein that was introduced by a collaborator. It's called octopus-like 2 from Arabidopsis. It has very few homologs. The sequence alignment is not informative in terms of conservation. And when you put it through AlphaFold, you get something that looks like this. However, we used looking at attention patterns to find actually that there is a previously unnoticed zinc finger motif. We saw that these C's, it's C3H zinc finger motif, are highly attentive to each other. And then when we took those amino acids and looked at them on the structure, we could see that it did form a new zinc finger that has gone unannotated by all other ways. So, and then looking further into this protein, we're able to find a novel repeat motif and potential dimerization domain, which is composed of 6 repeats of this particular pattern, either G or P, then R, then anything, then S, then anything, then D or E. And so I just put a glimpse of the protein here just to give everyone a test to try and find this motif. Even if you know it's there, it's not so simple to look for it and highlight it. But what the language model, and so now I'll reveal the answer to that. And so here I've in the sequence overlaid the G, R, S, and D of our motif repeated 6 times, and also an indication of kind of thinking how the language model is seeing it, these networks of attentions connecting these different amino acids. And so this is a protein. The sequence has been looked at for at least 20 years. I looked at the sequence for at least a year on and off, and did not manage to see this pattern. But in looking at attention patterns between these different amino acids, it became immediately clear. So we can see that deleterious mutations weaken attention networks. And so this is a loss of attention. And so I drank a cold drink, I drank a cold shoe. Since that's not expected, you're going to have less strong attention. And similar to like putting a proline in the middle of your zinc finger. And so this is a known deleterious mutation that causes loss of iron sulfur binding. And you can see a loss of edges above a threshold with the deleterious mutation here of cysteine to serine. And so based on this concept, we are developing a new method for computational deep mutational scanning, where what we do is computationally vary every amino acid position, then measure disruption across attention networks, and then we can group patterns by the disruption pattern, looking for sets of amino acids that disrupt similar attention heads within the model. We can also look for mutations that are the most disruptive to various attention networks. And so that was how we found these two new motifs in this previously pretty unstructured looking protein. Um, and then we can also examine amino acids that cause major disruption to attention networks, which is allowing us to prioritize functional sites. So as a test case, I'll show MTHFR. Mutations in this protein affect folate reductase activity, which can lead to birth defects. And so of the top 10 MTHFR amino acid positions that most disrupt attention networks, the first is just the initiator methionine. 7 are known to cause folate deficiency or are in characterized substrate binding sites. One of them isn't found in human populations, but is the most disruptive in the experimental DMS of MTHFR. And the final mutation is immediately adjacent to folate binding site. And so what we're doing now is working on scaling up this analysis to many more proteins and many more characterized sites. So, and the key points of this is only about 50 to 100 human proteins have experimental deep mutational scans. This computational DMS can be run on a protein in just a few minutes, and we're developing it as a general purpose mutation affected motif prediction, where your input is just a single protein. And it's usable for any protein. And we're also looking at how different language models are kind of prioritizing different sites. So as the other broad base, I'm asking the question of can pre-trained embeddings capture protein function? So the most powerful demonstrations of applications of protein language model require additional training on labeled data sets. This is the great work in structure prediction, protein design, drug target interactions, antibody engineering, etc. But there's kind of a disconnect between machine learning and what goes on in a lot of biological research. It's a persistent mismatch issue, is that biological researchers often have specific and focused protein problems and do not have large data sets, which really excludes them from a lot of the really powerful tools of machine learning, which require large labeled data sets. And so what I investigated, or what we investigated, is can we use the pre-trained PLM embeddings directly to represent protein function without having to do additional training? And then does embedding similarity actually reflect subtle functional similarities and differences? So this is the promise of function-aware embeddings. We want protein embeddings that can faithfully reflect true functional and structural properties that can be used directly with fine-tuning, and where mutations to these proteins will cause perturbations to the model that are similar to the scale of perturbations to the actual protein. And so there's a difficulty in this, in that data sets documenting subtle but real functional differences between closely related proteins are very rare. And what we need is a gold standard data set for evaluating subtle functional differences. So it's been broadly shown that these pre-trained embeddings can cluster proteins by class or by fold or enzyme specificity. But we want to know that when two embeddings are more similar to each other, that means that two proteins have more similar function than two that are further away. And so what we settled on was cross-species complementation assays as a strict test of subtle functional differences. And in these experiments, what happened was essentially remove an essential yeast gene and see if a corresponding human gene allows the yeast to grow. And so I focused specifically on these triplet cases, where first we have a yeast gene, it's been knocked out, there's no growth. We can replace it with one of two human paralogs. A2 human, when replaced, does not complement the missing yeast gene and causes no growth. But A1 is able to replace the yeast gene and lead to some growth. And so what we expect is that the human protein that's able to complement the yeast protein will have a higher embedding similarity to the yeast protein than the non-complementing protein does. And so we found that embeddings can be better than sequence similarity at prioritizing the complementing protein. And this is work with our two graduate students, Rob Shaw and Sam Love, which is currently in review. And so what we see here is that ESMC embeddings from both the 300 and the 600 parameter model are able to outperform pure sequence similarity at prioritizing which of the two human proteins is going to replace the yeast gene. And critically, they only exceed the performance, the accuracy of sequence similarity when instead of summarizing them with mean pooling of the amino acid embeddings here, we instead summarize them with pooled slice Wasserstein embeddings as introduced recently. We can also look at the case that ESMC does not get right by plotting sequence identity versus embedding similarity. And in red, we see the mismatched case, and you see it's quite subtle. So I expect as language models improve, we're going to get better at this task. And so what this does is, also another feature for using just straight embeddings. If we look specifically at the ESM2 series of model, you see that accuracy in this task improves as the models get smaller. And so this is something that has also been reflected in zero-shot prediction of fitness landscapes. And kind of one of the theorized ideas is that the larger models are memorizing extra information, and that's affecting the embedding similarities because what's being captured in the embeddings is also some irrelevant or artifactual stuff. The smaller models are forced to generalize in that they can't memorize every single thing about a protein sequence, but that instead forces to learn more general properties, which makes them better at this task. And so ESMC as a multimodal model, this additional, even though it is a very large high parameter model, the other modalities beyond protein are likely working to suppress this memorization because the memorized features are not going to be interconnected with the true biological properties. So we were very happy to find this ESMC embedding, which gave us this kind of first confidence that embeddings directly from a pre-trained model are capturing subtle functional differences. And this enables us asking a larger question. How do we distinguish between the many similar proteins within highly expanded gene families? And how can we see what makes structurally homologous proteins have different functions? And so there are many highly expanded gene families in humans, which are sets of proteins that are structurally pretty similar, but have some type of different function. And so our motivation is asking, why do two similar proteins have different functions? Now we're framing this is if we can find the mutations necessary to convert one protein's function into that of another, we can understand really what makes them different. And so this was kind of a classic question in biochemistry of enzyme conversion. And so kind of a flurry of papers in the late 80s and 90s of converting trypsin into chymotrypsin through mutation. But it was actually a relatively rare case. This is a very hard rational design challenge, changing enzyme specificities. And so our goal now is to convert proteins' functions to match another's in a few mutations as possible. And what we're doing is using the just pre-trained PLM embeddings as a proxy for functional similarity. And so in one case study, we can look at major spike protein G. This is a protein responsible for host selectivity during bacteriophage infections. If you look at the overlay of spike protein G from two different phages, G4 and Phi-X174, you can see that by eye, there's nothing that you could really clearly look at and say, that's the difference. Like that's the amino acid that I would need to change to make this selective for the other host range. And so I'll start with introducing this technique. What we do is we randomly mutate the G4 spike protein G into the Phi-X174 spike protein G over many iterations, which are shown in these gray lines. in order to understand the correlations between different mutations. We then use that information to determine an optimal mutation order with the goal of increasing embedding similarity as fast as possible. And yellow is our final generation. And so through this example, you can see an interesting feature of linked mutations where embedding similarity never increases until all 4 of those linked mutations have occurred. Even in our best path, we have a period of no improvement until the critical mutations are made, and then we have a rapid increase. In some of the random trials, you also see there's one random trial where all 4 mutations, the last of the 4 mutations, wasn't made until the very end, and then we have the boost. And those mutations are plotted here. And you can see that 3 out of 4 of them fall onto this one data sheet. And so I've kind of this vision for interconverting protein functions. First, to study species-specific protein differences, particularly in the case of failed cross-species complementation. Like, can we rescue some of these failed complementations of the human paralog into the yeast gene by doing just a few mutations? Another potential application would be converting a mouse protein to more closely match a human protein's function. So instead of deleting the mouse protein and putting in the human protein, can we directly humanize the mouse protein? It also is an opportunity to say, like, okay, I'm using a mouse model. Is the protein that's already in the mouse close enough? And if not, what are the mutations that are differentiating it from the human protein? And we can revisit these classic biochemistry problems of interconversion between enzyme specificities. And then at a larger scale, because this is a relatively fast process, we can look more broadly at the sequence to function question and systematically test which types of mutations across many species, many proteins are driving these functional shifts. Like, what type of mutation tends to be the ones that are going to give us a big boost in embedding similarity. And then say one of those mutations is characterized to be disease causing, but another has no information. Maybe we can use that to prioritize that unannotated site. And so I'm specifically studying highly expanded human protein families with a focus on GPCRs to test receptor specificity conversion. And so, kind of in review of the talk, you can think of protein language models as a new toolbox for understanding proteins. We have this model that's learned a lot of things about the structure of valid protein sequences, and we can use that to directly study protein sequences. So think of just the past few things that have happened in the past years and months on predicting structure and predicting interacting proteins and predicting drug and small molecule binding. But with directly looking at just embeddings from the pre-trained language model, we can prioritize these functionally important sites by looking at sites that the model cares about. We can reveal structural motifs or other co-evolving sets of amino acids. We can explain which amino acids differentiate proteins with different function, and also study functional differences between proteins at scale. And so as a last part that I didn't have time to go into, we also introduced a new technique to align proteins with very low sequence similarities. It's leveraging protein language models for accurate multiple sequence alignment. And interestingly, this is a new algorithm that shares no algorithmic steps with traditional ways of doing sequence alignment. And so instead, what we're doing is clustering amino acids based on their embedding similarity. So I wanted to thank everyone for listening and coming to this webinar, and also mention my lab members, Robert Shaw, Samuel Love, and Ahmad Shamal. Thank you.
Thank you. That's fantastic talk. Let me ask whether Eric has any question or comment before I'm asking some question.
Well, thank you. I think that was a great talk. Thank you for presenting it. I love the way that you're able to use the language models to predict, structure, function, and activity, as opposed to the traditional things that I used to do in grad school. So seeing that done in a practical way, I find really fantastic and fascinating. And love to see how this is going to be impactful on for companies, and what your next stages may be in translating this into something going forward. Any thoughts on that?
Um, Not really. Maybe I'm too deep in academia. I haven't found anything specifically commercializable, but I'm open to it.
But to me, I mean, once you kind of figure out how to routinely predict function of the protein just based on the sequence, I believe a lot of people wanted to invest that technology for sure.
Yeah, when you're thinking about, to continue on what Taesook was saying, a lot of times there's evolution of proteins to optimize function. And especially if you're thinking in the therapeutic space and in the drug discovery space, and even if you're talking about synthetic biology to optimize, output of engineering, strains and enzymes to create a compound or something along those lines. If you can optimize them using something like this in a predictive way, then I could see a lot of applications for that as well.
Yeah, we're trying to match some of the results of directed evolution to see, okay, we know kind of the start and we know the end. Is there a faster path potentially? Um, yeah,
Eric, do you have any other? Oh, go ahead.
oh no, I just like the idea of swapping functions between proteins, swapping specificities, swapping.
I see.
And
Eric, do you
even
have any other thing?
well, I was curious when you're talking about the mouse and the human proteins, as opposed to what is needed in order to humanize a mouse protein, or what do you need to uh, that also is going to potentially impact the immunogenicity of therapeutics. So I can think of lots of different applications of it there, and um, just like keep an open mind about how things can be applied. Because I think the real impact, yeah, not the real impact, but a major impact on benefiting society is like really getting this out there to have that being applied in ways that are going to have an actual benefit on practical things. And so this is my bias. This is coming out here. So I love the work that you've done, and I love to see where it goes.
Thanks.
Yeah, so the my question and comment also similar. Nowadays, a lot of people also funding agencies also interested in AI thing. And then that means that you have the a lot of opportunity in terms of getting funding, but at the same time, if this really work, I mean, just based on sequence, you just tell me, I mean, the function, I'm gonna buy that technology for sure, by paying whatever money. And then a lot of pharmaceutical company and a lot of other company also doing so. I believe so. The question I have is, during entire your talk, I keep thinking about evolution because of the language evolved, and then now we have the language, multiple different language. I in Italy, I feel like I'm stupid because I cannot speak in Italian. And then somebody just talking to Italian, I'm talking to English. We sometimes communicate, but not necessarily clearly. Uh, in terms of the protein evolution, the protein I mean, mutate randomly, and then selection actually uh, lead to the protein collection we currently have, right? But that's a random process. And I believe you are, because I understand some part, but I didn't understand the other part. I guess your, approach also includes some evolutionary process. And am I right? Or I kind of assume in a long way.
So I think the core evolutionary signal that's being captured is coevolution between
Mm-hmm.
acids, has been shown by other people, and also preference for amino acid. And so, like the kind of you can think of the tendencies of bacterial proteins versus eukaryotic proteins to be kind of two dialects of proteins,
Mm-hmm.
proteins that have the same meaning. And in the like pretty classic Bertology Meets Biology paper. They show the distribution of points for bacterial and eukaryotic proteins do have some um, separation.
I
So
see.
I think that's most analogous to the um, dialect thing and kind of
Hmm.
acid choices in bacteria for best way for being a bacteria or being a eukaryote. Um, Other dialect things is the rules of sequence for disordered sequence is different
Hmm.
rules of sequence for structured sequences.
Hmm.
And so I think there's potential interesting work at kind of separating out these two rule sets. In disordered, it's more about like regions of physical properties, and in structured, it's more about like we need to form a structure. Um, and so I think, yeah, that's kind of another interesting future thing. But for the most part, it's just interesting to study this kind of Rosetta Stone that can see these patterns and sequences that we can't see. Like when we look at a FASTA file of like 600 characters, like we really can't take almost any
Hmm.
near zero information from that.
I
But
see.
think of this model that's seeing all these patterns and all these connections between amino acids. It's a potentially a going to be a very useful tool for dissecting protein function.
I see. So the one more question is the from the pretty much kind of naive person. I'm not doing any language model thing. I'm just using the AI model. I mean, or the AlphaFold or Rosetta. I'm just, I mean, coming from that. So the one fascinating thing that I already mentioned is if that sequence functionality, prediction is possible, that's gonna be huge breakthrough, in my opinion. But I guess, I mean, you also mentioned some of the challenges during your talk, but what would be one major challenges we need to overcome to make that happen?
So there are some groups working on really nice tools to take a protein sequence and give like a plain text prediction of what the function is.
Mhm.
I think our current limitation in that is proteins that have no characterized function. Like the function that this protein does has never been seen experimentally, and we
Mhm.
and so I think the currently the protein function prediction is there's been some really nice things on the when a function, some other protein is known to kind of have that function or something analogous. But I think the big gap is these unknown functions, uncharacterized proteins in, random single-celled eukaryotes in the ocean have all kinds of proteins that no one's ever seen and no one's ever looked at. And so I think the big bottleneck in true functional prediction is what's always been the bottleneck is missing annotations. And so what can we do towards actually predicting new function and not annotation transfer.
I see. So
I
you
think
are saying
that's a challenge.
we
I don't
need
know
to have more experimental data to make that prediction better, or you are saying,
get more experimental data somehow on protein function,
uh,
or we can figure out a way to through well, like through these protein small molecule screenings, we can now do like fast um computational screening to see like, okay, what thing is going to bind this protein?
uh-huh.
Um, like this type of prediction, the dream is to be able to take a sequence and know, okay, this protein is going to react with these molecules. It's going to probably have these PTMs. It's going to
Mm,
probably interact with these other proteins. Um, but yeah, that's the dream.
I see. I see. Okay. Let me check. Eric, do you have any other thing, comment, question?
No, none at this time, man. It was really well done. Thank you.
Okay, so in that case, I mean, I wanted to just close. Okay, so thank you all for joining and staying today. We'll meet again on July 3rd, Thursday, the same time, the same Zoom link. We'll have Professor Rodrigo Ledesma Amaro at Imperial College London and Dr. Heidi Klump at Boston University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you. I stop recording. Just one second.